we all are touched by disease and by the frustration of not knowing, not able to detect, not able to characterize, and only at the beginning of treatment, really. That's where we are. And I think that to be in that interface is something that has been pure luck for me, and I'm going to squeeze it like a lemon until the end of my days.
Welcome to UnNatural Selection, a show about competition, innovation, and the strategies shaping tomorrow's frontiers. I'm Nick Encina. Each week, we speak with pioneers shaping the future.
Welcome back to UnNatural Selection. Today, we're joined by Jose-Carlos Gutierrez-Ramos, Chief Science Officer of Danaher, to explore how innovation is reshaping the life sciences, from diagnostics and biomanufacturing to AI and drug discovery. JC is Senior Vice President and Chief Science Officer of Danaher. In this role, JC is responsible for setting Danaher's science and technology strategy, supporting M&A and open innovation, and counseling Danaher's operating companies on innovation opportunities. He leads the Danaher Innovation Centers and the Danaher Scientific Advisory Board. Working with technical leaders across Danaher, JC enables innovation best practice sharing and culture building. JC has a PhD in immunology from the Universidad Autónoma de Madrid and trained as an immunologist in Spain, Germany, and Switzerland. He started his academic career as assistant professor of genetics at Harvard Medical School. He has authored more than 150 peer-reviewed publications. Let's go.
JC, thank you for being here. It's such a great pleasure to have you, and it's great to see you again.
Same here. It's great to be with you and your audience.
We go back 10 years. It's been a while, and
more.
I think the first time we made contact, you were a leader at Pfizer, and I think I just cold called you on LinkedIn just to make contact and just kind of start a conversation. And then over the years, we've been involved in startups together, and we've just remained long-term friends. So it's great to finally have you on the show.
Same thing here.
To get started, I always try to set the context and start with a foundational question that gives the listeners an understanding of, as we go through the course of an hour conversation, what has motivated you all this time, and what really drives the work that you do. So can you please let us know, JC, in your own words, what need or impact drives your work?
A profound, I've been a scientist like you for 30 years or 35 years, and I fundamentally love science. The process of, discovering one area and going deeper into it and learning about it, and the fascination, I use this word sometimes, I get transfixed in some of these fields that we just go into. And so that's what really, really is at the core of my personality and behavior. Really, is curiosity, is exploration. And I was lucky because I could have, and also maybe exploration, curiosity, and maybe a little bit of ADHD as well. I come from Spain. I was born in the 60s, so there was no ADHD, but it was like just a guy that was not focusing. And I was lucky that by chance through my life, I could put that passion at play towards trying to help human health. And so if I can say in one word, and I hope that this doesn't sound megalomaniac, I do it at my level, of course. And what drives me is that I've been lucky enough that I can use my passion and my instincts and my training to try to help, to have an impact in improving health outcomes. And also, it's very personal for me. When I come from a family of Navy sailors in Spain. Mm-hmm. My grandfather was an admiral. My father was this. My uncles, my aunts are married to sailors. So I was training to be a Navy sailor. And then I got a non-Hodgkin's lymphoma when I was 21. My spleen broke. My liver was a mess. And so, And I was a year in the hospital, literally, with chemo, and
Wow.
this was pretty Rituxan. And so, and that changed my life completely in terms of trajectory. And I always say I'm a patient. And for many years, I was a patient because, I was like sort of this submissive behavior that, you want to be good, you want to get through. And I saw many people in hospital and later on that were not as lucky as me. Lucky, really, because medicine, to some extent, is luck still. Less and less, with more molecularly driven. But imagine so it's, to make a long story short, that I think is a combination of a personal motivation, and we all have it. In my case, it's that one, but it's your parent, it's your father, it's your mother, it's your wife, it's your daughter. It doesn't matter. We all are touched by disease and by the frustration of not knowing, not able to detect, not able to characterize, and only at the beginning of treatment, really. That's where we are. And I think that to be in that interface is something that has been pure luck for me, and I'm going to squeeze it like a lemon until the end of my days. I'm not moving from there for that intersection of science, medicine, and awe and exploration.
Well, I think for the benefit of those of us that are recipients of all the medications and all the therapeutics that you've worked on, I think it's a good thing for us because you've been so influential. Again, I started by mentioning you were at Pfizer, so from the pharma space to the now you're in research and diagnostics. You've been in startups, and so you've had your fingerprints are throughout kind of the spectrum of from research to discovery all the way through treatment and so on. And so I think we're lucky to have you be so inspired by this. And then, just for the sake of the audience that may not be familiar with the space, can you let us know, for people outside the industry, at a high level, what does Danaher do, and what role does it play in the broader research diagnostics ecosystem?
Yeah, no, it's of course an important question. I'm more than happy to tell you this. Maybe I make an intro to that. In my career, as you said, through pharma and biotech, trying to develop drugs, I had two consistent barriers, even when I was accountable for them. I couldn't blame anybody. It was me. It was in my department. And I'm talking of other barriers and other obstacles than the complexity of biology and pathology and disease. That of course, goes without saying. But, you get at it. But there were two things. One was patient selection. You might have a drug that is active. And most of the drugs are not active, meaning that has efficacy. And you might not know, and it's active in 30% of the patients that you try. Today, unless you can predict what is the 30% of patients that you try, you might not develop it. You might leave it. Phase 3, you might not get funded in the last stage because it's difficult to compete. It's at risk. Now you're going to put another half a $500 million to $1 billion, depending on the indication, in that last push, and you don't know exactly who's responding. You might end up at the same 30% as a competitor drug. So that, or you get through, but then in the marketplace it doesn't perform well. So patient selection, super important. And I'm going to go back, of course, to Danaher. And then the second topic has been always a limitation, has been that to do true experimental medicine, meaning to experiment, of course, ethically and with all the controls, if a drug works in humans in early phases of development, is truly not doable at scale because it costs you $3 to $5 million generate enough drug to do a small experimental study. I'm talking about a naked antibody. I mean, a simple antibody treatment. And that, of course, how many of those can you do? So just to go back, then these two were barriers to really do experimental medicine, to choose the best drug. Or if I have a drug that maybe is not the best, but is good enough, to select the patients. And I reached the conclusion that pharmaceutical companies probably are not the best place to do either. And I tell you why. Uh, Precision medicine or personalized medicine requires detection, requires diagnostics. And in a pharmaceutical company, of course, you have a large division of what we call precision medicine colleagues that are finding the assays. Of course, we understand the pathways. They configure an assay, they deploy it to a clinical trial. But if you are lucky that the assay performs, then you have to partner to bring it to scale, not just to 10 clinical sites or to 100 clinical sites, but to thousands of clinical sites. You have to partner with a diagnostics company, and the diagnostics company incentives and the pharma company incentives are not necessarily aligned, and it's invariably a jerky advance. Mm-hmm. So I reached the conclusion that unless diagnostics companies really pick up the ball of evidence generation and on precision diagnostics, it's going to be very difficult to take advantage in real time of all the great drugs that are being developed. It will happen, but it won't. Instead of taking 10 or 15 years, it's going to take 20 to 40, because diagnostics companies have traditionally worked in a more technical comparison realm rather than generating clinical evidence. And Danaher, has a diagnostics division that is around $12 billion in sales. We're up there with Roche and with Abbott. We perform 1.6 million diagnostic tests an hour.
Wow.
In up to 40,000 sites, maybe 18,000 hospitals. So it has the scale to address that issue. Can we truly enable precision diagnostics? And I will talk more about that. So Danaher does diagnostics, and then Danaher in one division. In the second division, called biotechnology, also is called Cytiva, there's a lot of biomanufacturing. And to address the second topic that I was talking about, the ability to really lower the cost of drugs, the drug manufacturing, that allows much more experimental medicine or much more access even later on. And this is obvious, for example, in new modalities like cell therapy or gene therapy, as you know, is very expensive. But even in antibodies, again, most of CMC and manufacturing of antibodies is recombinant DNA technology of the 90s rather than synthetic biology of the 21st century. And I think because the pressure is so high on pharma in the moment that you have a signal in human, just to move the drug forward, even if the process is not perfect, it doesn't matter. At the end, it's 12 to 14% of cost. Cost of goods is only 12 to 14% of the cost of the drug. Mm-hmm. So it doesn't matter really. It matters, but it's not that dominant. So I think that companies like Danaher or other companies are in a better position to bring the latest science to biomanufacturing. And that's the second division that we have, and that's where I came here. The third division that we have is a life science analytical methods and reagents company that is also similar, around $10 billion in sales, from instruments like mass spec, microscopes, reagents like antibodies, conjugates. Overall, if you look at the three divisions, diagnostics, life science, analytical methods, analytical instrumentation, and biomanufacturing or biotechnology, we really are an enabler of biopharma globally. And that is what, and I think the unique opportunity to bring, and that's what, of course, drives us to work every day, the latest science and technology to accelerate each one of those.
Mm-hmm. Yeah, it's a great, thank you for that. And it's great that you touched upon the challenges that pharma companies have. Like yourself, I've been in biotech and pharma. I've been in life sciences and research. I've been all the way downstream to clinical delivery and point of care. And a lot of times you hear people that don't know better talk about, and by no means is are pharmaceutical companies saints, right? There have been tremendous errors made in the past by some companies and so on, like all other industries and companies. But you hear people talking about how drugs should be free, or you hear people talking about how pharma companies aren't incentivized to cure diseases because they would rather sell the treatments for it. And when I hear this stuff, I think to myself, like, maybe they should read a little bit more about the process. It's incredibly hard discovering drugs that work at these minute details and modifying whether it be a molecule, an antibody, or a gene, or any combination of things. And even if you can get to that point, it's even hard beforehand to understand what the mechanisms of action are in the first place, and be able to then manipulate them in such a way that you can save a person and not just be toxic and hurt them more. And there are so many complexities in between that everywhere that I've been, whether it's been helping to start biotech companies or consulting for bigger ones, everyone's always trying to do the right thing. They have their strengths, they have their weaknesses, but I agree with you that pharma companies aren't necessarily in the best position to be able to apply modern technologies, especially because what they're doing at scale requires a lot of their thinking, a lot of their concentration, following regulations, and all the other things that they do. So a lot of the experimentation can be left for the research and diagnostic companies. They're relatively a little bit more nimble.
Yeah, yeah, no, that's right. So, as you know, for example, I don't know the numbers in 2025, so I might be dated, but 2034, 3 out of 4, well, 2 out of 3, don't get me, uh, biologic drugs that were developed in pharma came from biotech. So the discovery, and again, same thing, very likely for small molecules, the discovery piece of it, the pathobiology, and even the platform, the therapeutic platform, right? I mean, cell therapy was started not in a big pharma or CAR Ts, or then it became big, but was really either academic medical centers or small biotech. So the process of bringing new targets with new candidate drugs and new platforms, other than small molecules or antibodies, even the antibodies started outside of what was pharma at the time in the 80s and the 90s, comes from smaller nimbler. But they rarely invest because it doesn't have the same value perception upfront on manufacturing optimization or on patient segmentation. And I think that is where, and as you said, there is a gap because when it comes to pharma, again, I'm focusing now on, again, on clinical development. There is going to be 90% of the cost and the risk that I'm going to take. So there are gaps in different areas that I think that have to be taken by some group. And I think Danaher is a perfect group to do it. And there are others, of course. I think it's a little bit of musical chairs at the moment. In how, particularly now with computing and data science that you know very well, of course, I think in the next 10 years, I feel like musical chairs, like everybody's up, there are 5 chairs, and who is going to take the seats? It's not clear because discovery is going to look different, obviously, much more computational. Even modeling that before, I mean, I'm talking about pharmacological and physiological modeling was like hampered. It was like one hand in the back. It was based on ones and zeros rather than now having a much more complete picture. Then, of course, clinical trialing that was really inefficient and will be improved. And so I think in this process of of moving chairs, I think there is a great opportunity for us as patients, for us as scientists and clinicians, to really have an impact.
Yeah, I agree. And I definitely, a lot of what you're saying resonates with me. The historically, diagnosing and treating disease, if we look back, just even human history, has been a random walk of luck. If we look way back, when we were treating disease with either herbs way back or even with leeches and bleeding. It was just random luck, and they had all kinds of names for diseases. And then sometime during history, we ended up coming up with names like cancer, which also meant a whole bunch of stuff because it was just like a general thing that makes you sick. Then we
Right.
realized, no, it's actually, there are different types of cancers. And then genetics came in and we realized, okay, even breast cancer means multiple things. And now we're understanding more that even those things can be multiple different diseases within one area. And so I'm really excited by it, like you said in the beginning, where the science is going. Now we're understanding far better, and there's still so much more to learn, but we're understanding so much at the multi-omics level, the genomics, proteomics, lipidomics, metabolomics, all these things. Independently, we're learning a lot. We're not crossing that border yet where we're starting to look at them in combination. And as you know, what bubbles up as your health is a combination of all those things, including your behavior and your environment and your socioeconomics. But now you start looking at very, very complex data. You don't really know what the relationships or the patterns are, but in walks in the modern technology of AI. AI has been around for a long time. We've been doing machine learning and neural networks for a very long year, but the LLM form that connects all these patterns. And so I feel like things are accelerating and things are going to get very exciting. But to your point about the musical chairs, I hadn't thought about it like that. But one of the things that I have seen just in this podcast alone, I've interviewed pioneers in the life sciences, in the pharmaceuticals, in the medical devices, and even in the big digital companies like
Right.
the Verily's and Microsofts. And one of the things that I'm noticing is there's this convergence.
That's
The
right.
the the borders between, just 5 years ago, there was a very clear separation. You're either life sciences and diagnostics, or you're pharma, or you're digital. And now they're all kind of like there's Venn diagram where they're all morphing more so. Where now I'm thinking like, oh wow, Siemens is a competitor to Thermo as much as it is to Sanofi. And it's like, it's not clear who's who. And so when you said the musical chairs, I started thinking to myself, like, yeah, it's actually happening very, very quickly. And it's not even just within one industry, it's a bunch of industries all coming together, vying for the same four seats.
That's right. That's exactly right. Exactly right. It's even more than before. And that's good for us. I mean,
Mhm.
it's good for society. It's good for, and I think this disruption that we see is, companies will get dislocated or dislodged. So two things, as you were talking, two things came to my mind. One, I think it's important also for all to realize, listen, of course, disease and drug development and drug discovery has been a little bit like connect the dots. You remember this game? I think it's called Connect the Dots in English, where you go and say, one dot, and because it was really binary, 1 to 0. Okay, do you have this assay? And when you get to the hospital or even in the lab, okay, do you have this protein? 1 0. What's up? Do you have this gene mutated? 1 0.
Mhm.
And so we had a sometimes few dots, and we have been adding dots, but at the end dots. And then we were, I think, until recently, and still today possibly, in a situation where you look at the dots and say, oh, gee, is this a horse? The legs, or is an elephant? Now, you don't see the trunk, but that is to some extent, certainly in my time. You're younger than me, but in the early 2000s, that's how we were doing drug discovery. This in 5 years, in 10 years, today we see it in language, as you said, and in coding, you know. But for biology, I think that before we know, we'll go from connected dot to see an image to a raw video data file of your disease.
Mhm.
Like this. I mean, again, I say this to shock my colleagues because that's the comparison. And of course, it will not happen overnight, but a full characterization. And of course, the problem is that we have been talking about metabolomics and genomics. I've been talking. I work for a company called Millennium Pharmaceuticals in the late 90s. And of course, we were dreaming with all this, but the technology was not there, the physical technology. And then the physical technology was developed, of course, with sequencing and this. But then the computing technology, as you said, was not there really. Now, and you know more than me, I'm amazed by the ability of autoencoders to transport data of different types. So I think that what you described before, maybe one thing that you have to validate or not because you are the expert, but I think that our ability to merge data, multi-layer data, that before it was not possible, or was anecdotal, one paper that compared this, but at a massive scale to generate that raw video file of your disease specifically, I think is closer than ever. Maybe not completely there, but close, don't you think?
Oh, I totally agree. To your point, the examples we have historically of connecting all these different dots multimodally has been for a paper where some people very painstakingly went through and they already kind of knew the answer beforehand. They kind of wanted to show that, hey, this works. And there's a science to be had in looking across layers. I think the exciting part now with the modern technologies is we can start to get to a point where it can be done at scale in clinical implementation. So it's no longer theoretical, some interesting paper in Nature. Now it's something that your clinicians might be able to use in real time. Not today, but in the next 2, 3, 5 years. And science is just going to completely change based on all this knowledge that we've been building since the Human Genome Project 20 years ago, all the omics, LLMs. I think all these things are converging. And I think part of the reason why all these different companies and different industries are converging too is because they see the opportunity. They're like, I don't want to miss that boat. I don't want to be the one standing at the end of these musical chairs.
Exactly. And how science, you know, this, I mean, we think a lot about this, of course, and act on it ourselves. With this 1.6 million tests a day that we generate. So, of course, there are chemistry, immunochemistry, genomics, I mean, PCR, pathology. So, when we look at disease, I'm going to start in diagnostics and then I go to life sciences. When we look at disease, unfortunately, we are biased depending on your training. Oh, I'm going to look at genomics, right? I mean, your genes, and I look at ctDNA. But of course, maybe the best, no, maybe not, almost 100% sure that the best definition of your disease is a combination of one chemistry, 10 genes, 2 proteins, because at the end, you don't know where you're going to, in that moment in time, other than having longitudinal follow-up of yourself, at that moment in time, maybe something that was a protein or is now a metabolite downstream, or something that was a gene, was not quite mutated, but is a post-translational modification that you wouldn't get. So I think very likely the multi-modal or the multi-parametric combination of diagnostics would be much more powerful in defining your journey. And similar in biology, I think this multi-modal definition of a process, the suppression of the tumor microenvironment of a tumor, right? Instead of looking at 10 things for the paper, as you said, not for the paper, that sounds bad. No, for a genuine interest that you have, but that you cannot scale, you cannot generalize. I think you guys, the computing guys have brought this important term that is general. I mean, I think it's coming from you guys, I don't know. Generalizable. These generalizable platforms that are physical or computational is super important. And I think that's where science, or at least biomedical science, got a little bit stuck. And a lot of the things that we were doing were not generalizable. And again, we couldn't, I guess. I mean, I'm not blaming ourselves. You had an observation, was reproducible, and therefore you put it forward. But it was not generalizable. I think that the scale at we operate today allows to really be more generalizable. Something else is coming to my mind. My daughter is making is a PhD in neuroscience. She's doing a PhD in neuroscience, and she took. I hope that she doesn't listen to this. She would not like it that I quote her, but she took a biostatistics course. No, the first 2 years of their PhD. And I go, oh my God, this is super important. And then she goes, Dad, this is completely outdated. And I said, no, I mean, because in parallel she's doing work, she's working databases, 10,000 subjects with imaging and this and that. And I said, well, then the examples, of course, in the book in traditional biostatistics are statistical analysis of, I don't know, 50 mice with this and that. So I think that we see a complete dichotomy in which new generation of scientists are all data scientists. In fact, I gave a talk recently where I was just saying, which science? You mean data science? Because it's difficult to separate both anymore. Remember how we, even in your time, I think it was like bioinformatics and then
yeah.
the other ones. And I think when I go to a company and they have this separation, and of course, some separation still makes sense, but it's a new world where data the, is our gate to this multi-layered, multi-model reality that will get us closer to the truth.
I totally agree. Yeah, it's and I think whether you consider yourself low data, big data, medium throughput data, ultimately the sciences are really all data sciences, right? You're just trying to collect as much data as you can. Sometimes it's little if you're working with cells. Like I was a stem cells 25 years ago, you collected very little data. I was still a data scientist. It was just like individual data points, or if you're
Right.
working with genomics, or if you're working with this with astrophysics, it's tons of data. But I think those barriers are starting to thin out now, because there is a lot more data and there is ways of dealing with it that I think, especially with the way that AI is disrupting a lot of fields, the people that are going to be the most well off in the future, regardless of what field you're in, are the ones that are comfortable dealing with a lot of data and making higher order decisions
That's right.
that are driven towards a science or society, things that impact humanity.
That's right. That's right. Even I mean, again, I don't want to go too deep on this one. If you want to go to another, just redirect me. But even, as you know, lab science is, and we deal a lot with this in our company, in our analytical instruments and reagents division called Life Science Instrumentation Group. Um, how is going to be the lab of the future? The lab of the future, I don't mean 100 years from now, 5 years from now.
Hmm.
What is the role of a scientist where 90% of the work that we do in the lab is very likely, you know, automatable, and agentic workflows can be taking care of most of it.
Hmm.
And also how much of the lab work will be data collection versus hypothesis testing, physical versus hypothesis testing, mainly computational for all the reasons that you said. No? Because probably it's more, because it's difficult in a physical world deal with all the parameters that you want to deal with when you ask thoroughly a question. And I think that world is something that we are at Danaher, we're very, very interested in. And we just signed for example, a big partnership with a company in London called Automata that is basically had this concept of a lab bench, like the ones that we always had, but it's not human. It's not just automation, it's autonomous, right? And it learns, and of course, there are different lab benches for different functions altogether, or different, uh, not just one task, but one discovery or development or, or process developments. But it's learning and it's going back and it's connected. So, what is going to be a scientist? And is the main creative science the one that is going to be is going to make questions computationally, and uh, and the other is going to be agentic workflows that just load, uh, you know, all the cell types that you can get with as many conditions as possible and measuring as many things as possible, and by multicellular combinations in organoids and tumoroids, just to get to another level of complexity, and then another level of complexity. It's going to be fascinating. And we are trying to position ourselves to help in all these parameters.
Yeah, that makes sense. So if we pull this back to Danaher, in
Mm-hmm.
in your role as CSO is largely to be able to steer innovation in the direction of the company. What does innovation mean to you in the context of the work that Danaher does? Obviously, innovation in pharma is a different, in software it's a different ballpark, but in the Danaher sphere, what does innovation mean to you? And then the second part of that is, how did your experience being a patient affect your thinking about what innovation means?
Yeah, yeah, very good questions, Nick. So maybe I start with the first one. Say, well, you know, one of the things that I do, and with my colleagues, to step back and say, okay, well, in a world like our world today, with all the vectors and variables and technology changes and this, what are the pillars of where biomedicine and biomedical research is going to go? Regardless of the how, the pillars are really four, I think. One, and we try to address them with our technologies and build them in one way or another, alone and with partners, right? But the four are, you want to detect disease early as possible, and you know the numbers, right? I mean, 4 weeks in the delay of a detection of cancer increase mortality by 10%. No, the detection, no, treatment, delayed treatment in cancer. 50% of lung cancers are detected at stage 4, small cell. In neuro, it takes 18 months to 4 years from the beginning of in neurodegeneration, the beginning of the cognition decline to get to a neurologist. And 70% of those patients, by the time they get to a neurologist, already have passed the window of treatment. And again, it's the beginning of it. Now there will be more treatments, of course, the beauty is that, but still. So detecting early, I'm not talking about screening. Screening is, of course, important, but we'll get to that. Just early detection. So that's one, and I will come back to that. Second, of course, detection early is important, and molecular definition of your disease, number 2, super important in cancer. What is your disease? What are the, as you were saying before, well, we call breast cancer many things we call, and now we know the triple negative. So what is your disease? And we're getting more and more in cancer, certainly better, but in other areas, autoimmune disease, not good. So number 2. Number 3, the treatments would be more and more tailored to your molecular characteristics. Now, 80% of the drugs that we develop in oncology are molecularly targeted. And it's not personalized to your, like we do it now in genomic medicines for rare diseases. It's not gene editing for one, right? And we are very big in that. We'll talk about that later. We have decided to build and we treated patients with a physician scientist for gene editing in vivo. So it's not going to be that way, but it's going to be more and more personalized. And if cancer vaccines makes it with personal neoantigens, if they make a big break, that will be a big direction. That one. So and this goes back to our patient segmentation before. It's going to become more and more personalized. And fourth, once you are treated and you are in remission and your disease is more and more chronic, and we have seen it, right? Multiple myeloma, 10 years ago, 15 years ago was a death sentence. Now it's a chronic disease. Breast cancer was a disaster. Now, still, you can be unlucky, but for the most part, you are managed well. HIV, you name it. So more and more of these, deadly sentences are now chronic diseases, and therefore, detection of relapse, we call MRD in cancer, but in general, monitoring yourself and understanding what your trajectory is going to be super important. So these 4 things define us as patients, because it's important to us, define how medicine will be, probably broadly, but also define the type of research that that we're going to be doing in academia, because at the end, you are asking one of those questions directly or indirectly. And so at Danaher, we're trying to address these 4. And in early detection, for example, we have, I would say, one of the most potent platforms for immunoassay. We detect atomoles of a molecule. We detect in the periphery, and again, we publish on this, neurodegeneration proteins that other people can detect only in the CSF. We detect it in the periphery. Post-translational modification. We launched a machine called DXi 9000 that is really remarkable. We bought Abcam. Abcam is an antibody company that antibodies allow us to detect this type of analytes just to increase our menu. and give those. So this is at the protein level. At the genomic level, of course, circulating tumor DNA has been fundamental, and we have several companies that have partnered with Exact Sciences, with Natera, that build the libraries. A company called IDT that build libraries to detect your tumor. So super, how do you call it? Super sensitive detection is important, and then it will be complemented by this computing that we were talking before, multi-layer. In understanding molecularly your disease, one of our companies, and there are others, but I think we're very good, called Leica Biosystems, a pathology company that is really trying to determine in the tumor simultaneously, in a quantitative fashion. I'm talking about generalizable. Of course, we can detect, this molecule from the tumor microenvironment, this molecule from the tumor, this molecule from the immune system. These are the few players that determine if you are going to progress or not. Well, now we have technologies that allow us to quantify. And of course, a pathologist or a doctor wants a clear-cut report. They don't want, you have a little bit more of this. And that is something that is not easy, but we'll get there. Mm-hmm. We and the field will get there. Because, it's important to say, we both have bladder cancer, but the best target for me is the ADC called Nectin-4, and for you is combined with immunotherapy, and for you is FRNA2 with something else. I think we will get there, and the field will get there too. As there are more and more precision-targeted medicine, you need precision-targeted diagnostics. And then in therapeutic tailoring, I think for rare diseases, N of 1, N of 100, N of 1,000, a lot of it is going to be mRNA-based gene editing for true genetics. Then I think mRNA vaccines. I'm not talking about prophylactic vaccines. I believe in those too. I mean, obviously they don't give you long-term memory, but it's a question of doing research. We'll learn how to do that with other modalities. So we'll learn to do that. But just CAR Ts are now being done in vivo with mRNA transfer. So there's a lot that can be done. And then going back to computation, even for small molecules or for antibodies, the fact that you don't take 2 years and 10 cycles of medicinal chemistry and synthesis and optimization, but rather a month to get your candidate, because computationally you can generate trillions, right? Mm-hmm. Also makes the possibility of having a much more tailored to your polymorphic. Remember, your genes are polymorphic, mm-hmm. and therefore your target for disease X might be slightly different than mine. Mm-hmm. And, if in a month with relatively, or in a week, with relatively less effort, I can get a drug that is for your polymorphic variant, and you and the guys that have your genetic configuration, that's what we're going to get. And then finally, the last thing that I say is this following disease trajectory is super important. And, we just announced that we acquire a company called that we would like to acquire a company called Massimo. We haven't closed the deal yet. That is a company that has specialized in sensor technology, particularly around pulse oximetry, but many sensors. And we believe that also the whole area of sensing, in addition to biochemical and genomic, I think will bring another level of longitudinality to your disease trajectory or patient trajectory to determine, is the disease coming back? Are you likely to relapse? Are you not likely to relapse? How do you move? The expression of your face, the color of your skin. There's so much information, as you know, mm-hmm. that probably by itself would be enough, but certainly for a while combined with biochemical parameters. So I think these are the 4 areas. The 4 areas, and these 4 that guide our direction, to some extent, are the areas that as a patient, I would like to see. For years, I was terrified of the disease coming back. And even today, if I don't know, I get a bad flu or something, I call the doctor right away. So I think that to understand that your disease is coming back, understand, am I going to be 30 years? If somebody would have told me, hey, you know what? You had this mutation, and this is your blood profile, is unlikely. I mean, you have 0.1 chance that over the next 30 years you have a relapse, or that you have your. I will have lived the first 5 years after I left the hospital much better than later.
Yeah,
Um,
sure.
so anyway, I think these are the key principles. I think in moments of change, we have to uh, and we have to take advantage of the change, 100%, embrace it and lead it, but at the same time anchor to what is important for us as humans, right? Particularly in health and disease.
Yeah, I totally agree. And yeah, like we said in the beginning, both of our careers have spanned kind of like the full spectrum from early discovery down to point of care. And so you said a couple things there that really resonated. It's sometimes you're so focused on the silo that you work in, which is like some scientific discipline or some technology, or it could just be a device, that you don't think about how it will be adopted into the broad population. And you mentioned there, a clinician, when they get a report, they want something that's actionable. I've worked with so many clinicians and they tell me it's like, okay, your technology is great, but don't come to me with more problems. I want
Uh,
solutions. So if you're going to identify a risk, if you're going to identify some biomarker, whatever it is, your report better tell me what to do. And then I'll take it from there. But if you come to me with some statistical model that tells me there's a risk here and there's no solution, nothing I can do about it, I don't have time to go and do the research and figure out what to do with that. And then all the way down to the level of patient care, like you said, as a patient, one thing is to treat your condition. Another thing is to answer some fundamental human questions that you may have about what does this mean for me going forward? What does this mean for me, my diet, or for my children? Any number of things that more times than not, patients end up leaving the doctor's office with kind of more questions than answers, and very unsatisfied and very scared. And so the more that we can kind of take the initiative of thinking how to solve a particular scientific problem, but then also how it will be implemented in the course of care, and then how it affects all the stakeholders downstream. I think that uh, I think that science eventually is going to move down that path, especially as we have more people like us that think at a broad scale, thinking more than our little window of work, but really how is this going to affect healthcare in the long run?
Not only people like us. I mean, I think people like us for sure, but also, my wife has developed this interaction with a language model around the health of our dog. I mean, I'm using this where that language model knows the history of the dog, knows my wife, and suggests very sophisticated, not therapy outcomes, but therapy solutions. But when my wife said, "What if I do this about a test or a treatment, or could this be this?" There is quite a learning that has occurred about the specific individual. In this case, my dog, and I use it on purpose versus a human. But you know that that I'm surprised. So I use this as an example to say we as patients, all of us, certainly some of us have the privilege of education and experience and this, but all of us have more and more access to knowledge,
Hmm.
not just a website knowledge, but rather a learning evolution knowledge that will get to know more about us and about the questions that we ask and the level that we understand. And that is going to help us all as society to really
Hmm.
be much more fine-tuned about what we expect from care,
Hmm.
right? Um,
I totally agree.
yeah,
So, JC, this has been fascinating. I could go on for another 5 hours because I love hearing from you. I love learning from you. But we started with kind of what motivates you. And
yeah,
throughout your career, you've accomplished so much, and you're still doing so much. And so, if we zoom out for a second, just to finish off with the zoom out question, and think about a point in time in the future where you're ready to reflect and look back at all the work that you've done currently, and even things that you anticipate, things that you're planning on doing in the near future. What would be a best case scenario that where you could look back and feel like you achieved the fullest impact that you were capable of? What changes in the industry or improvements in human health would make you the proudest and feel like you achieved that deepest sense of fulfillment?
yeah. The bar is changing for all of us, right? I used to think, okay, you know what? If at the end of my career when I moved from academia to industry, I thought if I participate or help develop one drug, that would be good. And that would be enough, no? And certainly I've been lucky that over the years, we developed 4 drugs that are in the market and are helping, hundreds of thousands of patients. And then you think, well, of course, and that's super important. But now in this new world, which you can imagine all the things that we have been discussing around, what would it be? I think that, I go back to one topic that we discussed. I think that we will change fundamentally the attrition rate, mainly the failure of drug candidates, and would enable that more personal, less toxicity, more efficacy, would be enabling this, what we call experimental medicine, or translational medicine, we call it at times. Experimental medicine sounds, tricky. I don't mean it. I would say ethically, but really say, well, for your disease, for my disease, what is the best drug? And with all the safety coverage that one can have, to test it in me. And for that requires a much faster and precise ability to generate these clinical probes or candidate drugs. And that, I am certain, this would make the continuum of drug development make a quantum leap. And that could be mainly physical, but eventually would be also computational. Because, the more you measure in a human after giving a drug, the more you can model it, etc. So I think that to enable that truly personalized, to be part of the true enabling of personalized medicine, both from generating the drug and testing it quickly, will be my dream, I hope. I mean, it's being tight for me because I'm aging, but I have another 5 or 6 years to give it a shot, so I will try to help. Thank you, Nick. This has been a pleasure for my side. Thank you.
Oh, it's been wonderful for me, and at the pace that science and technology is moving, 5 years is an eternity. So I hope to have many more of these conversations, maybe working together someday to try to solve this problem, because it's near and dear to my heart as well. But JC, it's been a privilege knowing you all these years, and an honor getting to know you and having you as a friend. Thank you for being on the show, and I look forward to many more conversations.
Thank you so much, Nick. Thank you. Good luck. Ciao, ciao. Bye-bye.
That was JC Gutierrez, and now, UnNatural Signals.
The following are key takeaways from my conversation with JC. Number 1, the biggest breakthroughs often come from solving bottlenecks, not inventing new ideas. In drug development, the limiting factor isn't a lack of scientific ideas, it's the cost and difficulty of testing them at scale. The same pattern appears everywhere. Innovation accelerates when constraints on experimentation are removed. Number 2, entire industries stall when critical partners have misaligned incentives. Pharma needs diagnostics to scale precision medicine, but their incentives historically diverged, slowing progress. Many industries have similar friction points where collaboration is essential, but incentives aren't aligned. Fixing the alignment unlocks growth. Number 3, when measurement improves, the entire competitive landscape changes. Better diagnostics transform how diseases are defined and treated. In any field, improved measurement and data visibility reshape strategy, competition, and what success even means. And number 4, the next wave of innovation happens at industry boundaries. Life sciences, data science, and technology are converging into a shared arena. The same pattern is unfolding across sectors. The most important innovation opportunities now live between disciplines, not inside them. Thank you very much, and we'll see you next time.
Thanks for listening to UnNatural Selection. If you enjoyed the episode, subscribe, leave a review, and share it with someone shaping the future. Explore more at unnaturalselection.net and connect with us on LinkedIn and Facebook. Until next time, keep evolving.